William Caterall is a professor and chair of the Department of Pharmacology at the University of Washington, where he has been a faculty member since 1977. He received his BA in chemistry from Brown University and his PhD in physiological chemistry from Johns Hopkins University. He was a postdoctoral fellow and a staff scientist at the NIH for a few years before moving to the University of Washington. Dr. Caterall and his colleagues discovered the voltage-gated sodium and calcium channels responsible for generating the electrical impulses necessary for most physiological functions. His lab continues to study the structure and function of these channels, their physiological regulation, and their interaction with medically important drugs. He's also interested in understanding how impaired channel function may lead to human disease. Please welcome Dr. William Caterall. Watch step. Do you want to hold this or have it mounted?
I'll put it on there because I need two hands to
Okay, no problem.
change slides and point.
Let me get you your slideshow.
All right, perfect. So thank you very much for that kind introduction. This is my first periodic paralysis meeting, and I have to say I'm very pleased to be here to learn from all of you about this family of debilitating and difficult diseases. I appreciate the work that Linda Feld did as an organizer and in organizing my trip here. I also appreciate very much the beautiful lecture that Professor Steve Cannon gave earlier this morning, introducing you to the physiology and the pathophysiology of periodic paralyses. So in my lecture, I'm going to focus on the sodium channel as a molecular signaling machine, and I hope I'll show you how it works normally and also how its function is impaired by two specific mutations that cause hypokalemic and normokalemic periodic paralysis. So this is very much a basic science lecture, but I'll do my very best to explain as I go along, to take advantage of the nice introduction from Professor Cannon this morning, and eventually we'll come to movies that show just how the channel works, and they'll be easy to understand. So picking up on the introduction of Dr. Cannon for just a minute, I want to point out that in nerve and muscle, electrical signals are generated by first sodium ions moving into the cell, which causes a depolarizing phase of the action potential, shown here, sodium moving into a muscle fiber, for example. And then within a short time, a millisecond or so, potassium ions move out of the cell, and that brings the electrical signal back to baseline and resets the resting potential. And as this slide says, all this work is done by ion channels. And it was discovered quite a long time ago by Hodgkin and Huxley in the 1950s that this is the way an action potential is generated. And this is just a slide from their work to show you that you can even look at a real science slide and understand all about it. So here's an action potential here, this line, rising phase, falling phase. So their work showed that the rising phase is caused by sodium channels, which open and then inactivate during this time, and then by potassium channels, which open and finally inactivate during this time. So when I began my lab in Seattle, and as was noted, it was a long time ago, 1977, our first aim was to understand what are the molecules in the cell that cause this electrical signal. And so for several years, we used neurotoxins, which paralyze their prey by altering sodium channel function, as sort of molecular fishing bait to dig out, to fish out the sodium channels from brain. Oops. Oh, I have to go backwards here. How do I do that? Okay. Sorry. Well, the problem is this came up, so it's not responding for the moment. Terrible to have a Mac user using a PC. I'm going to go back. One more. There we go. That's the slide I'm looking for. I have to be careful here. So we found that sodium channels from brain, and in parallel work from another lab showed this was true for skeletal muscle as well, are formed by a large pore-forming component, or subunit, as we say. So this protein, called the alpha subunit, forms the pore through which the sodium ions move. And though we didn't know it at the time, and it's not on this slide, it also has the voltage sensing apparatus. So all the functional pieces of sodium channels are really in this alpha subunit, and that is the target for the mutations that cause periodic paralysis. So in my talk, I'll really only focus on the alpha subunit, though sodium channels have two other smaller beta and 1 and beta 2 subunits. So the sodium channel is a large protein. Now, many of you are not biochemists, so this won't make any sense, but it's a protein of about 260,000 kilodaltons. So that's a big protein, but even a big protein is a small thing. So the sodium channel is actually about 10 nanometers, and I want to get you used to thinking about small things, because we're going to look at single sodium channels. So a single sodium channel is 10 nanometers, 10 billionths of a meter. So imagine how small that is. And although the electrical signal in nerve or muscle is, we can measure it in millivolts, a single sodium channel makes a tiny electrical signal. And on the slide here is shown a single purified sodium channel turning on and off, making its little electrical signal, which we measure as 15 picoamperes. Now, you know amperes, right? You have 15 amperes in your circuits at home. So this is 1/trillionth of the current that's in your 15 amp circuit at home. So small molecule, small currents, but they initiate all the electrical signals in nerve and muscle. So what does a sodium channel look like? Well, it shouldn't be this hard, but it won't go forward. Oh, no, there it is. Okay, good. So as Professor Cannon showed this morning, sodium channel is made up of a long series of amino acids, 2,000 of them, which we sort of diagrammed as a string here. And that string folds back and forth across the membrane 24 times in making a sodium channel. Now, this is an exploded view of the sodium channel. This exploded view of the string actually folds up into a three-dimensional object that looks something like that. So this long series of amino acids, 24 transmembrane segments, is divided into 4 repeated parts. We call them domains. So repeated parts, or domains. You can see them here, 1, 2, 3, 4. Each one looks like the other. They're not identical, but they're very similar. And each domain has a voltage sensing module, the segments 1 through 4 here, and I'll speak about S4. S4 is this one, segment 4. That's the one that has the positive gating charges that Professor Cannon showed you are the targets for mutations in hypokalemic periodic paralysis, and will be a central feature in my talk. So then, in addition to the voltage sensing module, there's a pore forming module here that is the pore for sodium ions to go through and initiate the physiological activity of the sodium channel. And these 4 components, 4 domains, fold together to form a three-dimensional channel. Now, the beginning of the work that I'm going to tell you now began with the discovery by David Clapham, a leader in our field, of a bacterial ancestor of mammalian sodium and calcium channels. It's kind of a crazy thing, right? I mean, there's no nerves or muscles in a bacterium, and we don't know what this channel's doing there, but it's been very helpful to us because the bacterial sodium channel, diagrammed on the side here, is very small compared to this large and complex human sodium channel, but it has exactly the same core functional units. It has a voltage sensor, just the same, and a pore, just the same. And a bacterial channel, 4 of these molecules come together in an array to form a fully functional channel. So we've been able to do detailed chemistry and biology on this bacterial channel and learn a lot about the human channels that have evolved from it. So this is our first view of the bacterial sodium channel. And now you're looking at a single sodium channel. Remember how small it is, 10 billionths of a meter. So looking down on it, as a sodium ion would, there's the hole in the middle. So this is the physiological pore through which sodium ions normally move. It's formed by these blue voltage sensing modules that I showed you on the previous slide. And it's surrounded in a symmetrical way by 4 voltage sensing modules. So each unit, each subunit has these 2 functional modules coming together to form this tetrameric structure. So we stopped moving on the right here. What happened? Let me see if I can make that come back. There we go. Keep moving now. So here, the voltage sensing module on the outside in green, you can see it has 4 transmembrane units. The pore forming module in the middle in blue, and importantly here, the red linker between them, which I will start calling the S4, S5 linker. It's the linker between those 2 functional units, and as you'll see, it's quite important. So I want to focus first on the voltage sensor. This is the target for the hypokalemic periodic paralysis mutations. Good golly. So the X-ray crystallography eye view of a voltage sensor. Let me explain it to you, and I'll try and tell you some molecular landmarks that are important for the way it works. So you remember this had 4 transmembrane segments, and they're sort of hard to see here, but they're like this: the S1 segment, S2, S3, S4. But what this slide shows you are the gating charges. Remember, these are the targets of mutation. They're called, they're all arginine gating charges, so they go by the single letter R. There are 4 of them, R1, R2, R3, R4, arrayed across the membrane. And you can see their side chains which carry a positive charge. They're 1, 2, 3, 4. So there are 2 other important landmarks here that I need to introduce to you to tell you how this works. One landmark is called HCS. This has the term hydrophobic constriction site. Sorry for the long words. It's really the rubber seal of the voltage sensor. It keeps the water and ions on the outside or the inside. They can't go through here. And then there are 2 red areas shown here, ENC, extracellular negative clusters. So these are negative amino acid side chains here in red that interact with the positive gating charges. As you know, negative and positive like to interact with each other. So here it is at the single voltage sensor level. And on the inside, you can see the R4 gating charge now is inside the hydrophobic constriction site. It's interacting with the intracellular INC negative cluster. So on the next slide, I'm going to show you a movie of how this works, but I'll just kind of preview the movie by telling you what you're going to see. So this picture here is an activated voltage sensor. 3 of these key gating charges, R1, R2, R3, are on the outside of the hydrophobic constriction site. So these guys are going to move back and forth across the membrane in response to membrane potential. When you depolarize to activate the channel, they move out, and so this is the activated form. 3 are outside. Next slide that I'll show you the resting state, and 3 of these charges are 2, 3, and 4, in that case, are going to be on the inside. They will have moved in. Okay. So as I said, this little movie, I'll run it in a minute. But this is the voltage sensor over here. This is a pore. We won't talk about that right now. We're coming to that soon. Here are the gating charges. They're in blue again, but now 3 of them, 1, 2, 3, are on the intracellular side of the hydrophobic constriction site, identified here by this green amino acid. So this is the resting state. 3 are inside. In the activated state, 3 are outside. And now, if we're lucky, we'll get this movie to work. There we go. Out they go. See that? So when the cell is depolarized, those gating charges move out. Out they go. And one more time. Watch as they go out that they pull here and they open the pore here. But we'll see that in more detail in just a moment. So one more time. Resting state, activated state, back to the resting state. Okay, so looks pretty simple, doesn't it, when you look at it that way? Electric field exerts an electrostatic force on those charges, just like you learned in high school physics. And they move either out or in, depending on whether it's depolarization or hyperpolarization, repolarization. Okay, let's look at the pore. So you've seen this before. And here the blue is the pore-forming module. So here's what it looks like with just 2 of the pore-forming modules shown on the 2 sides. And the gray here is the water-filled pathway through which sodium ions move through the normal physiological sodium channel. They go through a narrow spot here called the selectivity filter that chooses sodium, doesn't let the other things come through. At a central cavity, which is a rather large space in the channel. And then, crucially, for what I'm going to show you next, the activation gate is formed by the intracellular, that is the downward end, of this S6 transmembrane segment. So this is sort of the pore lining segment you can see here. And it's tightly closed. There's no space here. And that's because this is the closed form of the sodium channel. So we're going to go back to our movie, but now we're going to be looking at the movie from bottoms up as if you're sitting inside the cell. And you're looking up at the sodium channel, and I'm going to show you how this opens to let sodium ions through. Okay, another movie. We always have a prayerful moment just before we hit the movie. So when I hit the movie, you'll see here, this is the gating, these are the gating charges. They're going to move out, so away from you, toward the back of the slide. They're going to pull here on this S4, S5 linker, which I've introduced, and here is the closed pore, not much space, and they're going to open that up. So let's see, what do I do? There we go, just like that. And it doesn't look like it makes much space, but it makes enough space for a sodium ion to go through. So first the voltage sensor, then pulling here on this blue segment, watch the way it moves, and then this opens the pore to let sodium ions in. There we go. And then, of course, the pore closes because you have to have multiple action potentials, so sodium comes in, then the channel closes. Now, the channel has one more trick that I have to show you. So in muscle, as you've heard, when you strongly contract your muscles, there are many nerve action potentials coming at high frequency, and then there are muscle action potentials that drive the contraction of the skeletal muscle. So for the nervous system to work, you have to be able to fire at high frequency, so it isn't enough for a sodium channel to open once, right? It has to open, and then it has to close or inactivate. And then it's ready for another trial and another action potential. This slide shows you that we also know how the channel inactivates. And it's a bit complicated, but not too bad. So same thing you've seen before, it's the pore. What the inactivation process does is it makes a partial collapse of the pore, which I've tried to diagram in this slide, and then I'll show you a movie of that too. So if we look at the selectivity filter, this is the way the normal selectivity filter looks. It's nice and square, and sodium ions go through very well. In the inactivated state, that changes so that it gets narrow. So essentially, 2 sides have collapsed in. As you can see here, it looks more like a rectangle or a parallelogram. Same down here at the activation gate, the bottom where the channel opens and closes. In the resting and open state, it's kind of symmetrical. It makes like a circle. Whereas here, you can see two of these pore lining segments have moved toward the center and blocked the pore. And you can see it a little better down here. So closed, no space. Open, this is what you saw before, lots of space. Inactivated, it's closed. But if you look carefully, you'll see this is a square, and this is not a square. It's a rectangle. So this is asymmetrically closed in the inactivated state. Okay, so back to movies. We're looking up again from the inside of the cell. And when I run the movie, actually two things are going to happen. This is the resting state. Notice that this is square. It will open, then the channel will be open, and then it will inactivate and close, but the closure won't be square. It'll be a rectangle. So I hope you can see that. It's a little bit subtle. Here we go. Okay, open, inactivate. Now notice that that is different from what we started with. I'll try and go back to the beginning. Not exactly sure how to do it. There it goes again. Yeah, sorry, I can't go back to the beginning easily here. So you'll have to look very carefully. When we snap back, this will go back to the square configuration of the resting state and then open. But I think it'll be hard to see. Okay, square, and then open, and then inactivate. See that? Okay. So those are all the tricks of the sodium channel: resting, open, and inactivated. Okay, so now I want to turn to the disease part. So again, this slide summarizes things that have mostly been discussed very nicely by Professor Cannon, but just want to re-emphasize them in context here. So, of course, periodic paralysis is the episodic flaccid paralysis of skeletal muscle, which you know well. It's caused by mutations in these two distantly related ion channels, NAV1.4, CAV1.1. I would emphasize that the bacterial channel we study is the ancestor of both of these channels, both sodium and calcium channels in humans. And then 2 points, not so much emphasized before. The hypo-PP mutations are always in the R1 and R2 gating charges, whereas a more rare condition called normokalemic periodic paralysis, or normo-PP, are in the R3 gating charges. And these papers from Professor Cannon's lab and my lab worked this out several years ago. So let's think about what these mutations are going to do. So now I'm going to give you another slide that's familiar by now. It's the same old movie you've seen before. So in the resting state here, the R1 and R2 gating charges are right here at this narrow spot in the channel. They're actually preventing water and ions from leaking through the voltage sensor. The side chains in blue that are shown here are pretty much clogging up the space, so nothing moves through. Now, the mutations that I'll tell you about are mutations in these side chains which make them small. And when they're small, you actually create a hole, or at least that was the hypothesis, because this isn't here anymore, so there's a hole, and the hole is big enough for ions to go through. So now, using the magic of structural biology techniques, which I have to say I didn't invent, I just used them, we're going to be able to see the hole through which the pathogenic sodium leak occurs. While I have this here, I want to make the other point that this is a resting state, right? So R1 and R2 are here blocking. But now, when I run this little movie that you've seen before, what's going to happen is that R3, this one, is going to get up here in this space. And so hypokalemic periodic paralysis creates the leak by mutating these gating charges. Normokalemic periodic paralysis creates the leak by mutating this gating charge. And it only gets in the path, so to speak, in the activated state, right then. And so you get the leak in the activated rather than the resting state. And that makes the difference, pathophysiology of the disease. A little biochemistry, not too much. I want to point out that the normal arginine that is in those gating charges here is a pretty big amino acid residue, and you don't need to worry about the details of the chemical shorthand here. You can just see that it's long from left to right, and it has a positive charge at this end, as Professor Cannon pointed out this morning. So when you mutate arginine to glycine, you get this. It's much smaller, and it no longer has the positive charge. So that's the genetic lesion that causes the disease. Now, we'll come back to the fact that what you release, what's missing, is a molecule called guanidine. But keep that in your mind. We'll come back in a couple of minutes. You would guess when you take all this away by making this arginine into glycine, you'd make a hole. And so we set out to find that hole. We put the hypo-PP mutation into the bacterial sodium channel that we could study by X-ray crystallography at high resolution. Now, the first thing we had to do, and I apologize, this is a thick slide. This is for the electrophysiologists in the audience, of whom there might be 2 or 3. The first thing we had to do is we had to show that that mutation had the same effect on the bacterial channel that we knew well it had on the skeletal muscle channel, the human channel. And forget about the left side of the slide. That just shows that the mutant bacterial channel still works. The key part is the right side, and here. And again, relying on the very nice description from Professor Cannon, and now that you are a well-educated physiologist, you can see the leak current is small for the wild-type channel, but it's much larger for the bacterial channel into which we have put the human hypo-PP mutation. And it's larger, the leak is larger in the downward direction, and that means, if you look at the voltage axis here, the leak current gets bigger as the membrane potential gets more negative. This is a resting state leak, right? Resting state is present at the negative membrane potential around resting membrane potential. So the bottom is a normokalemic periodic paralysis mutation. We call it R3G. This is the one I'll show you the picture of, so you might keep that in your mind. Again, you can see with the naked eye, so to speak, that there's an increased leak, but now the increased leak is upward going, right? Not downward going. That's because the increased leak is in the positive membrane potential domain here. And this is where the channel's activated. So this is a leak in the activated channel. All right, I know that was tough. We're getting out of this physiology stuff right away. So back to looking at structures, and in this slide, as I go through, you'll see the hole that's made by that mutation of arginine to glycine. But first, let's start with the wild type and see what the normal channel looks like. So this is another kind of shorthand way to show the voltage sensor here in this panel. And you can see the R1, 2, 3, 4 gating charges. And in this case, we're focusing on the R3 gating charge right here. And it's mutated from the large arginine to the small glycine. Oh, I'm sorry, this is wild type. It's mutated down below to the small glycine, sorry. So wild type channel, this is a view of the top of the voltage sensor, and it just looks like a Martian landscape. Don't worry about it. What it intends to show is there's no hole here. You can't see through the membrane at all. Down here in the mutation, we're making this mutation, arginine 3 to glycine, G, right here. And look, Mom, we have a hole. So there's a hole that goes all the way through the membrane, through the voltage sensor, created by this one single amino acid mutation. So there's another way to represent this from the side. So this is a top-down view, as I suppose is clear. So top-down view. How about from the side? So here we're using a computer program which looks at these kinds of structures and says, where could water go in this structure? And so the kind of pinkish color here shows you where the water can go, and this is the wild-type channel. So water can reach in partway, but it can't go through. It can reach in partway from the bottom, but it's sealed. This is that hydrophobic constriction site. This is the rubber seal in the voltage sensor. So that's disrupted when we make this R3G mutation. From the top, you see the hole, and from the side, you see now the water-accessible pathway goes all the way through the voltage sensor. So a sodium ion can enter here and merrily go right through this water space, just as if the voltage sensor weren't there. And that's only true in the mutant. In the wild type, there's a gap, it can't get through. So we're also interested to look at that's a normo-PP mutation. We're interested to look at a hypo-PP mutation. Here we have to resort to molecular modeling because we haven't yet achieved the high resolution needed to see the hole directly. But molecular modeling is pretty powerful and it can illustrate what's going on. So again, closed gating pore here, in this case. So being closed by one of the arginine gating charges, and then looking at the model down here, again, we're looking at the water-filled space. In the wild type, of course, the water-filled space is not continuous. In this hypokalemic periodic paralysis mutation, in the resting state, the water-filled space is continuous. But now the R3G mutation, the one we looked at in the last slide, does not give you a continuous gating pore or continuous water pathway in the resting state, only in the activated state, as I showed on the last model. So this modeling fits exactly with the structures that we have and says that a hole is formed of these dimensions that allows sodium ions to go through. For a hypokalemic periodic paralysis, it allows them to go through in the resting state. So let me go on for another several minutes. Is that okay? Where's our organizer? I've done about 30 minutes, but in 5 more, I can do a little more. All right, carry on. Uh-oh, it's chemistry again. Remember, I told you to take a look at this. This is guanidine. It's actually a separate chemical. It's not an amino acid. It's not even a biological chemical. It's just a chemical. But it's what's missing when you mutate arginine to glycine, right? This is missing. So it was reasonable to think that, hey, maybe guanidine can go through a mutant voltage sensor in the same way that sodium does. And I should point out that guanidine has this positive charge that used to be part of the arginine. It's over here. So we just wondered, what if we put guanidine outside the cell? Would it go through the mutant voltage sensor? Would it go through the hole? And actually it does. Another physiology slide. I'll try and not spend too much time on it. So here again, and now you guys know how to read leak current, right? Wild type. This is guanidine going through this hypokalemic periodic paralysis mutation, R66G. Lots of gating, what we call gating pore current, lots of leak. And you can see here you get more and more and more leak conductance as you add more and more and more guanidine. So in a way, this is an experimental curiosity, and so guanidine can go through, who cares? So I'll tell you why I think we should care. So maybe not so much on this slide. This is maybe another who cares slide. So we asked if we could get guanidine to actually bind and stick in the hole in the mutant voltage sensor. It should, after all, there's space for it, because we just made that space by mutating the arginine residue and giving it the space to be there. And sure enough, and now this will give you a sense of how high resolution this technique, X-ray crystallography, is. We can see the individual guanidine atoms in there, and they're very small, and maybe you can't see them in the back, but there's a little speck of yellow right there, and right there, and right there, and right there. That's one guanidine molecule sitting in each of the 4 voltage sensors that have the mutation. Now, I don't have it on the slide, but the normal voltage sensor, the guanidine doesn't go there because there's no space for it. Well, so if you can put guanidine there, it means that you might be able to make a drug molecule with guanidine as part of it that would sit there and block this hole, you know, just like a stopper in your bathtub, and maybe that would be therapeutic for periodic paralysis. So that's an idea we have. We haven't pursued it very much, but we have 2 bits of evidence that I'll show you on the next 2 slides that give us some interest in this subject. So first, on this slide, we can see, as I've mentioned already, at very high resolution where the guanidine is, we can see exactly the chemical interactions that it makes in this binding site. They're shown here by the dotted lines, and chemists would know how to interpret that, but you can tell that we can determine exactly how it interacts with its surrounding regions of the channel. It's very high resolution. The mesh here is actually the experimental data from electric crystallography, and you can see that the mesh exactly fits this little drawing of the guanidine molecule, so you don't really have any doubt at all about what's going on there. In fact, it's such high resolution that we can see a molecule very similar to guanidine. It's called methylguanidine. It just has one more atom in it, and we can see that extra atom. It's right here, and you can see the electron density that extra atom contributes. It's right here. You can see this is elongated at this end, whereas this is not. So why am I telling you all this? Well, drug companies use data like this to design drugs to fit in that space exactly, a process called structure-based drug design. So we're interested in that. We have one experiment that's helpful. So we screened about 50 compounds from a friendly drug company, Pfizer, with an eye to see these ones they already had. They didn't really put much effort into this except allow us to use them. And we just looked to see if we could find anything that would block the gating pore current. And so a compound called xylylguanidine, I won't show you the structure, actually has that property, although it's certainly not a drug. It reduces gating pore current quite significantly from the black line to red when it's carried by sodium. And same thing when it's carried by guanidine from the blue line to red here. Unfortunately, this is a very large concentration of this drug, 5 millimolar. You won't know what that means, but it's a much higher concentration than is typical for drugs. So this is my last slide, and I just sort of leave with the hopeful note that it's at least theoretically possible to create such a molecule that would work at lower concentrations, block gating pore current, and perhaps provide relief from periodic paralysis during episodes, or even conceivably be used in a preventive way. Now, I want to wrap up here by just putting up a slide showing all the people who really did that work. You know, I'm just the spokesman. And here they are. And as I say, I won't mention them except for one person. My colleague Ning Zheng in pharmacology is a structural biologist, which actually I'm not. So Ning and I have worked together on all of this work that involved the structural biology, and his input is absolutely necessary to make this happen, and then all of the other colleagues have done the hands-on work. Thank you very much for your attention, and I'd be delighted to answer your questions.
Any questions?
Wonderful talk, Bill. I thought Steve Cannon down here under the bright light. Sorry.
Yes, I can't see a thing, but just go ahead.
I'd ask a question. Because you have such beautiful structural information of what we're dealing with, and you better than anyone else know that there's added complexity for the sodium channels in humans compared to the bacterial ones, because each of these voltage sensors is slightly different. So do you think we're going to be lucky enough to have a magic bullet that might work for several different gating pore leaks, or it's going to be really personalized medicine, one bullet for one mutation?
Well, I don't have a really good answer to that, but I think that we won't find one bullet for all of the hypokalemic periodic paralysis mutations, but I think we could find a bullet that would act on several of them. Those that make the large arginine residue small, I think would be susceptible to the same kinds of blocking agents that I illustrated here. But the drug discovery process is a long and difficult one, and as many of you will know, drug companies are not very effective at developing drugs for orphan diseases because they see no profit margin in selling the drug at the end of the line. So I haven't been able really to get drug companies interested in this, but I remain interested, and perhaps someone will pick up the thread and try it out.
I think I understood what you said clearly, but my question is, with the guanidine, what about when you don't need it? Is that a risk that you're taking a drug that is putting that and plugging that hole when there really isn't a hole?
Yeah, that's a very good point. So if we had such a drug, you could think of two ways that you might use it. You might use it in sort of an instantaneous sense when you have the impression that you're going to have a periodic paralysis episode. For example, if your periodic paralysis is triggered by exercise, as we heard this morning, and occurs just after exercise. I think it would be possible to have a drug that you would take at the beginning of your exercise and that would carry through and prevent the pathologic leak opening up at the end of your exercise period. You could also imagine having a drug that you would take all the time. I think this is a much more stringent criterion because such a drug might have many side effects, as you point out, on other physiological processes that would be deleterious over time, and it would be much more challenging for drug companies to find such a drug and get approval for using such a drug. But I think a drug for acute use might be helpful to many patients. It might also be helpful in resolving periodic paralysis after it occurs in a kind of a difficult case. So that, I think, is an easier mark than a drug that you might take all the time.
So, my question is, as a clinical neurologist, we use a lot of readily available sodium channel blockers to treat myotonia, like mexiletine, and I'm wondering what is the molecular action of these drugs on sodium channels, and traditionally we don't use them to treat the paralytic episodes of periodic paralysis. So I'm wondering if your lab or any other lab has studied some of these available drugs as a repurposing project.
So that's a very interesting question and an important one to consider. So here in hypokalemic periodic paralysis, the sodium that is causing the problem isn't coming through the normal physiological pore, right? That's the central pore that I introduced. That is the mechanism by which the normal sodium channel brings sodium into the cell. And in myotonia and in hyperkalemic periodic, I should emphasize that, kalemic periodic paralysis, paramyotonia congenita, the sodium that's causing the problem is coming through the central pore. The central pore, the normal pore, is open too much. And the existing sodium channel blocking drugs are aimed to block that normal central pore of the channel, and they do so with varying efficacy and with voltage dependence, and they're very good therapeutics. They're not helpful at all in hypokalemic periodic paralysis because the sodium isn't coming through the central pore. The sodium is coming through the voltage sensor, and it's depolarizing the cell, again, as Professor Cannon described. That depolarization inhibits sodium channels because they get into the inactivated state and they can't open and work. And so a sodium channel blocking drug of the traditional type would actually make hypokalemic periodic paralysis worse, and one should not use it.
Great. Any other questions? I'm in the back. I can't see. You have to raise your hand if... Oh.
Moving in a different vein from the microscopic level to maybe macroscopic or in vivo.
So I'm having trouble hearing you. Could you speak a little louder?
Yes. Moving
you.
away from the microscopic concepts, what do you think would be the outcome of stem cell research, or is in my case with permanent fibrotic atrophy of some of the muscles, allographic muscle transplant to bypass the S4 linker or the R1, R2 gate issues?
Well, I have to say I'm not really competent to answer that question. So I'm not a physician. As you may have heard in the introduction, I have a PhD from Johns Hopkins, but I don't have an MD from Johns Hopkins like Steve Cannon. So you should ask Steve that question. Yeah, I'd rather not comment on the clinical aspects. One thing I would say, if one could prevent the repeated episodes of paralysis, either by the kind of drug that I'm talking about or some other means, I think it would ameliorate the downstream adverse effects that are caused by cellular damage in the muscle and degeneration of muscle fibers. So I think it would be possible, if you can prevent the primary deficit, that these secondary deficits would improve on their own. Of course, since no one's been able to test that idea, we're not sure that it's true. But the downstream effects in skeletal muscle come from the fact that the muscle is living its life, the muscle cells are living their life with the wrong ion gradients, and that has pathologic consequences over time.
I mean, I will say we're not there yet with stem cell therapy to replace muscle, or even, the idea of like a muscle transplant. I've never heard of it. Is that a thing? Can you do a muscle transplant? So there's work going on, I'm told, but it's definitely an unmet need. And when there's a problem and it's significant enough, the humans usually find a solution eventually. So I think it's just a matter of keeping pressure. And that's part of what the PPA is, maybe to focus research on those areas that need discovering. So.
Yeah, I'll just add on to that stem cell therapy, at a basic science level, is a very promising approach for this kind of disease in muscle. As probably all you know, there are satellite cells, precursor cells that are ready to grow into new muscle fibers when the muscle is damaged. Of course, in someone with hypokalemic periodic paralysis, those satellite cells have the same deficit as the muscle fibers themselves. But if one could create stem cells that would work as muscle satellite cells and fill in the damaged areas with fibers that have normal sodium channels, then I think that's a good way to forward for the future. And there are a number of centers, including one at the University of Washington, that are interested in this kind of stem cell therapy. So I think one can hold out hope for that, but not anytime soon.
So just, I guess, a theoretical question, and then maybe we'll stop. And this is just maybe ignorant. That all the cells have, like a good copy and a bad copy of the channel. And so it's the bad copy of the channel that's like causing problems. So you can imagine if you put a muscle transplant, if you could imagine the thought experiment, that with that muscle, let's say you get a new thigh muscle, and now you have a total body paralysis, that new thigh muscle probably wouldn't be paralyzed, right? Or...
Well, you'd have to rely on the muscle fibers that are formed by the new cells. The reason that hypokalemic periodic paralysis is as serious as it is that the impaired sodium channel, the genetically damaged sodium channel, the mutant sodium channel, actually impairs the function of the normal sodium channel. It has what geneticists call a dominant negative effect. So how does that work? Well, the sodium leak that we've talked about quite a lot, and you've seen in various forms here, including the hole in the mutant sodium channel, that leak lets sodium into the cell. It increases the normal low level of sodium inside the cell from, say, 5 millimolar to 15 millimolar or something like that. And that depolarizes the cell membrane. So instead of having a nice normal resting potential at minus 90, a hypo-PP muscle fiber might have a resting potential of minus 65 or minus 70. So what does that do? That engages the inactivation process of sodium channels, which inactivates not only the mutant sodium channels, but the perfectly good ones, because they're supposed to inactivate it if they're exposed for a long time to minus 70 millivolts. So you end up preventing not only the function of the damaged sodium channel, but also the function of the perfectly good sodium channel right next to it in the cell membrane, because you've reduced the membrane potential, and that channel goes into the inactivated state as it is supposed to do. So for that reason, you can't really recoup the function of the good sodium channels in the same cell that is expressing the bad ones, because the membrane potential is wrong. You'd have to hope that the satellite cells that I mentioned would make new fibers in which the majority of channels would be normal and they would function normally. So we can all look forward to that. I'm even guessing it will happen someday, but not that soon.
Great. Thank you very much.